right, hello. A few years ago, was tasked or charged with the idea of trying to find a way to mitigate resistance. So not just identify it or determine mechanisms, but explore ways to reduce herbicide resistance in the field. And so that's what I'm going to talk about. I'll probably start out just kind of reviewing the goal, then talk about some of the advances outside of our lab or worldwide this past year, and then talk about what we've been doing, and then try to put it all together. So this area of genetic biocontrol, a little different than what we're used to with herbicides. So most herbicides are going to target some type of protein, okay? There is another area of biocontrol that I'm not going to talk about, and that is targeting not at the protein level, but what leads to the protein, which is the messenger RNA. And then what I'm going to talk about today is targeting, and there's different genes we can target, but going back to the actual gene before the expression takes place. So the idea is, and this is very simplified, and here I'm going to talk about it in terms of reversing herbicide resistance to susceptibility. It doesn't have to be that way. It can be targeting genes that can suppress the populations directly. So if we have a resistance gene and a susceptible gene, and we're trying to figure out a way to go back to susceptibility here, takes more generations than this. This is simplified, obviously. How would we do that? And so I'm going to talk about a gene editing way to do it. And basically, so this would be, if you heard of CRISPR-based gene editing, that's what this is, but it would be in the population, not in a laboratory. And the way gene editing works is that we have to have these two components. One is the protein, Cas9. There are other proteins that will cut a gene that we target it to, and then the guide RNA here brings that protein to the gene to cut it. So we're destroying a gene, and it's very gene specific, and so I didn't put the A's, G's, C's, and T's up here, but just try to get the idea that this is very specific. So it's species specific and gene specific. Then the way gene editing works is that once this is cut, then the cell will try to repair it. And in gene editing, you want to give it a template and say, repair it this way. So it's kind of like remodeling a room in your house. You need to destroy what's there first. And then you have your plans for I'm repairing it this way, okay? So that's kind of how gene editing works. And so in probably about 2014, Kevin Esvelt at MIT is the one who proposed using this system for pest control, including weeds, and I think Palmer amaranth was his example of all, although we didn't know as much about resistance in Palmer amaranth at the time. But what one needs to do is we would want a susceptible gene, and this is very, very simplified. Then a gene that we have to put into the plant, specifying what other gene we're targeting, in this case, the resistance gene. And then secondly, a gene for the enzyme that destroys the gene that we're going after. And then they come together, and here we destroy specifically the resistance gene. And then one way would be to edit it back to susceptibility. That really doesn't work in plants, and I'll talk about ways around that here in a bit. So the way we would kind of want gene editing to work in the wild population would be like this. So now we have destroyed our gene, and let's say that it's a herbicide resistance gene. And then we give it the template to repair it to, which would be susceptibility, and then we get susceptible gene. So we've gone back. And again, the guide RNA and the Cas9, they're the workhorse, so they need to be put into the plant. These are bacterial, so the plants don't have them. They need to be engineered into the plant. Now, plants don't like to do it this way. So when you cut a gene, you target the gene, and you cut it and destroy it. Plants don't like to use your instructions on how to repair it. Other organisms do it much more efficiently. Plant kind of haphazardly likes to just put the gene back together, and often it makes it non-functional. So that's kind of what we have here. So instead of getting back to susceptibility, we end up with a non-functional version of the gene, which is okay. So this is, if this is a herbicide resistance gene, now it's not functional, so it's not providing the resistant enzyme, okay? But this doesn't allow good propagation in a wild population. So that's the problem here. So within this last year, a couple of groups have, and really their strategies are pretty similar, at least for what I'm going to talk about here. Group in China, so the Yang Liu in Chinese Academy of Sciences, and also Jackson Champer at Peking University proposed what they call a toxin antidote system. And it more specifically is CRISPR-assisted inheritance utilizing NPG1. NPG1 is a gene that's necessary for pollen development. So if you stop the expression of that gene, it's not going to produce viable pollen. They did the work in Arabidopsis, which is a model organism. It's self-pollinating. It's not going to be a weed we want to target. But because they didn't want to go right to a weed that's going to produce seed and pollen, because these are genetically engineered, it's a little safer to use a self-pollinated plant in Arabidopsis. And I'll put these together on the next slide here, but a very similar system, Cleave and Rescue. So this is the Caltech, California Institute of Technology strategy. Their target gene, YKT61, also in Arabidopsis, is important not for just pollen development, but also ovule or egg development in the Arabidopsis. So the strategy is this. So instead of trying to target the resistance gene, leaving the susceptible alone, and also getting your whole guide RNA and Cas9 construct, which does the work, into the plant time and time again as the system is propagated. The strategy is this. You engineer whatever target gene you're going after. Now, could be herbicide resistance, but they didn't work with that. They worked with pollen development and ovule development. You make it very, very different so that your system of cutting the gene doesn't target that. And then you just let it destroy, in this case, let's say it's herbicide resistance, both the susceptible and the resistance. You don't have to worry about trying to discriminate that. And then what rescues the plant, so this is the antidote in the Chinese system, this is the rescue in the Caltech system. That gene, which is not cut because it's quite different, will rescue the plant and allow it to propagate even though you've destroyed both genes over here. So that's basically the system. The efficiency here is really important. So matching the guide RNA, which specifies the target gene, matching it with the target gene is really, really important. All right, so now what are we doing? So we're working with water hemp, and instead of using Arabidopsis, which we don't care too much about, as a self-pollinated system to keep genetically engineered plant contained, we're using tissue culture, which is not going to produce seed or pollen. And one of the things we want to do is work towards being able to test the efficiency of this match. And so, first of all, we have been able to do the gene editing of the ALS. So we're working with the group 2 ALS acetylactase synthase gene. We've been able to change it just as kind of a demonstration of gene editing. In this case, we start out with a susceptible yeast and go to a resistant resistance. So the idea is, obviously, we would want to go back the other way in the field. But this is just a demonstration of how we do it in yeast. Now, within water hemp, we need to get the machinery to do the gene editing into the cells. And so we developed the system, we're working on it, where we remove the cell wall. Now, this is in tissue culture here. Remove the cell wall, degrading them with enzymes, and so we get what are called protoplasts. So you don't have the cell wall, and so you can get your gene editing machinery into the protoplast a lot better without that cell wall there. All right, so we tested, now this is to target acetylactase synthase as a resistance gene, and just because we know a lot about that gene, so it was a good place to start. We tested different guide RNAs against that. That's the component that targets the gene. We tested one of 4. We got good targeting for one of the 4 guide RNAs in isolated water hemp DNA, but when we tried it with the protoplasts, we didn't get good targeting, and we couldn't get repair of the ALS gene, even in the haphazard way that plants do it, putting the gene back together again. We were able to get, and this is the Cas9 that is labeled, so you can see it within the protoplasts. We know we can get the components into the protoplasts, but those protoplasts just were not healthy enough. And so we took a look at oxidative stress, and here, the fact that these protoplasts lit up very green, that indicates that they are under stress. So it's something about removing that cell wall in these cultures. And these enzymes that remove the cell wall are from plant pathogens. And so there is a pathogen response that the cell gives, that it's under stress. And so we wanted to reduce this stress so that we could test the system, gene editing system in the plants. And so this was kind of our breakthrough. So I don't use the term breakthrough very often, but in this case, I think it's appropriate because we had tried a number of different things to get the water hemp protoplasts to have this reduced oxidative stress and the ability to do the gene editing so we can start testing this system in water hemp. And a graduate student, Peter Bierbauer, used, now this is, he tried many different things. This is the one that worked. 2-aminoindane, 2-phosphonic acid, AIP. What this does, it inhibits one of the enzymes that is involved in stress response in the plant. The pathway produces phenolics, which we just need to reduce that in order to have healthy cells. And so here's our protoplast, and here we can see we get division. Now, we had not gotten cell division from these protoplasts at all, and that is something we would want to do, and it's an indication of health. But with the use of the AIP, we've been able to get cell division, and up to a few divisions, and that's what we have right here. So there are probably about 8 cells or so in there, and this is in an agar gel, so it's harder to get a clear picture. But This indicates cell division. So now we have cell division and healthy protoplasts. We can go back and try the transformation of the cells and work on this matching our guide RNA to target genes. So the next steps, we'll probably still work with ALS as a well-understood gene. Try the targeting again in the protoplasts with the AIP, and also try to get the cultures to regenerate from single cells. There's good reasons why we would want to do that. And then also extend this research to Palmer amaranth. Right, and then to acknowledge my research specialist, Robert Saba, and then my graduate student, Peter Bierbauer, funding agencies, and also my Hatch project.